This lecture will focus on the different genes regulating the development of each of the basic structures of a flower. Floral development models were first built from studies in Arabidopsis thaliana and Antirrhinum majus or snapdragon. Based on studies on both these species, the ABC model of flower development was put together. The ABC model of flower development is a scientific model of the process by which flowering plants produce a pattern of gene expression in meristems that leads to the formation of a flower, the key organ necessary for the sexual reproduction in plants. The ABC model of flower development has two basic principles. The first principle is that distinct organ identities are specified by the expression of the ABC genes. Different combinations of these genes result in different whorls of organs. Let's take a closer look at what organs these ABC genes control. We have here a picture of the Arabidopsis flower, which is composed of the sepals, followed by the petals, the stamen, and the carpel in the middle. Remember that these organs or plant parts are arranged in concentric circle or whorls, such that if we look at this flower from a top view, the first whorl will be composed of the sepals, the second whorl will be composed of the petals, the third whorls will be composed of the stamen, and the last whorl will be composed of the carpel. Now the first principle of the ABC model states that there are three genes, genes A, B, and C, controlling the formation of these whorls. These genes can act individually or in combination with each other to produce or specify a floral organ. The expression of gene A gives rise to the first whorl, which is composed of the sepal. The combined expression of genes A and B gives rise to the second whorl, which is composed of the petals. The combined expression of genes B and C gives rise to the third whorl, which is composed of the stamen. Finally, the expression of the C gene produces the fourth whorl, which is composed of the carpel. The second tenet or principle of the ABC model states that A and C genes negatively inhibit the expression of the other. If you notice in our ABC model, we don't have any combined expression of genes A and C. Gene A can interact with B, and gene C can interact with B, but we don't get an interaction between genes A and C. And as I already mentioned, the reason for this is that genes A and C inhibit each other's expression and negatively affect the development of associated organs. For example, if A gene expression is missing or reduced, C gene expression can expand, resulting in more overlap of C with B and thus more stamen and carpel, but no sepals and petals. Conversely, if C gene expression is missing or reduced, A gene expression can expand, resulting in more overlap of A and B, and thus more petals and sepals, and no carpel and stamen. The loss of function of one or more of the ABC genes can result in homeotic mutations. Homeotic mutation or transformation refers to the conversion of one organ to another type of organ. To illustrate what homeotic mutations look like, let's look at a normal Arabidopsis flower here in the left. This flower has all the basic structures including the sepals, the petals, stamen, and carpel. In the right, you also have an Arabidopsis flower, but in place of sepals, we only have petals. The conversion of the sepals into petals is an example of a homeotic mutation or transformation. To better understand how loss of function in the ABC model can result in homeotic mutations, let's look at different case scenarios of loss of function in any of these genes. Based on the ABC model, we know that B is required for the identity of petals and stamen. Now, what would happen to a flower if the B gene is not expressed? Without the B gene, we will be left with only the A and C genes. We know that the A gene controls the formation of the first whorl, which is composed of the sepals. We have that whorl here. The second whorl is controlled by the combined expression of A and B. Since we don't have the B gene, we only get another expression of the A gene. That means we get another whorl of sepals instead of petals. Now the third whorl is controlled by the combined expression of the B and C genes. But since we are lacking the expression of the B gene, we only get the expression of the C gene, which controls the formation of the carpel. For the last whorl, we have the expression of the C gene coding for the development of the carpel. Missing the B gene function results in homeotic conversion of petals to sepals and stamens to carpels. So we end up with a flower composed of sepals in the first whorl and then carpels in the second whorl. This is an actual example of flowers in Arabidopsis lacking the B gene. You can clearly see the presence of the sepals in the outer whorls and then the carpels in the inner whorls. This is another example of mutant flower lacking the B gene. This is a snapdragon flower. Again, you can only see the whorls of sepals and the whorls of carpel. How about losing the expression of the C gene? The ABC model specifies that the C gene is necessary for the identity of stamens and carpels and for inhibiting the overexpression of A gene. What would happen to a flower that lacks the C gene expression? Without the C gene, we will be left with only the A and B genes. Also, because the C gene is absent, the expression of the A gene is expanded. In this scenario, the A gene will control the formation of the first whorl of the flower, which is composed of the sepals. The second whorl is controlled by the combined expression of A and B, which is responsible for the formation of petals. So far so good. We have both the sepals and the petals. Now the third whorl is controlled by the combined expression of B and C genes. But because the absence of the C gene allowed the expansion of the A gene, we get another combination of A and B, which provides another whorl of petals. And then for the last whorl, we again get the expression of the A gene, which will provide a whorl of sepals. With the missing C gene function, we get homeotic conversion of stamens to petals and carpel to sepal. So we end up with a flower composed of repeating whorls of sepals and petals, which might look like this. This is an actual example of Arabidopsis flower lacking the C gene. You can clearly see the repeating whorls of sepals and petals. This is another example of a mutant flower lacking the C gene. This is a snapdragon flower. Again, you can see only the repeating whorls of sepals and petals. What if the flower loses the expression of the A gene? The ABC model specifies that the A gene is necessary for the identity of sepals and petals and for inhibiting the overexpression of C gene. What would happen to a flower that lacks A gene expression? Without the A gene, we will be left with only the B and C genes. Also, because the A gene is absent, the expression of the C gene is expanded. In this scenario, the expansion of the C gene expression will give us a first whorl of flower composed of a carpel because that is what the independent expression of the C gene controls. The second whorl is controlled by the combined expression of B and C genes. This will be responsible for the formation of a stamen. Now the third whorl is controlled again by the combined expression of B and C genes, and this will give rise to another whorl of stamen. For the last whorl, we again get the expression of the C gene, which will provide another whorl of carpel. With the missing A gene function, we get the homeotic conversion of sepals to carpels and petals to stamen. So we end up with a flower composed of two whorls each of the male and female reproductive organs. This is an actual example of Arabidopsis flower lacking the A gene. You can clearly see only the presence of the carpel and the stamens and none of the non-reproductive parts of the flower. Studies on other plant species show the absence of the A gene. This indicates that the function of the A gene in controlling sepal formation in flowers might be less conserved across species. Additionally, missing the A gene function can also cause bud formation where petals would normally form. This is a mutant Arabidopsis flower missing an A gene. A diagram of a cross-section of the flower shows buds in petal position. The rightmost picture is a diagram of a normal Arabidopsis flower. These findings in Arabidopsis indicate that gene A can also function in floral meristem formation, which may be the more conserved aspect of the A gene function. The roles of the A gene in sepal and petal identity may be more limited to Arabidopsis and close relatives.